Thank you. Braulio, thank you very much for all you and your organization are doing to help our science connect with the world. We really appreciate it, and I think you will have understood when you heard our Vice President speak how privileged we feel to work at this university that has supported us over the past 15 years to try and build what I think is a useful model for other places on our planet. And it looks like our graphics have gone to sleep, so I'm now down to a 2-minute presentation. Help. Ooh, that's not a good sign. Well, I mean, what I want to speak to you about, and I think this isn't news to anyone in this room, we are working in... This is like, there we go. Beautiful. Thank you. We're working in a science where the subjects of that science are disappearing. There are not many sciences that can say that. Astronomy can't say it. Particle physics can't say it. And Braulio, you raised the point that we don't have an inventory of species on our planet. We certainly don't. But actually, I don't think that's very interesting. That's just the beginning. That's like having a thermometer and saying, okay, we could read temperature. That's not good enough to have a thermometer. You actually need to use it to go out into the real world and measure temperatures and watch how our planetary environment is changing. Similarly, for biodiversity scientists, it's not just the list of species. It's being able to go everywhere on our planet and read those species. And this is going to take a massive scientific enterprise, the like of which our science has not been involved in. So as we look back at the International Barcode of Life project over the last decade, I think it's provided some useful instruction, but it's the beginning point. It's the foundation for a very large program in research that I like to think about that's going to make it possible to address those goals of an inventory of species and being able to track species on our planet in a way that's absolutely remarkable. So I mentioned the fact that this notion of extinction, the loss of the life on the final frontier that we're embedded within, it's recognized everywhere. Pulitzer Prize-winning book this year by Elizabeth Kolbert on the 6th extinction. It's been 650,000 centuries since life on our planet was decimated, and we're living in the century where it's happening again. And I don't think history is going to be very kind to biological sciences if we simply sit back and watch it happen. This isn't just the popular press. If you go into the American Museum of Natural History, the Hall of Biodiversity makes it clear that the scientific community recognizes this. If you read a recent issue of Science, you see Mark Urban predicting that 1/6 of all species on our planet could be extinct by the end of this century. That's 1 species per hour. It's absolutely staggering. There have been calls to actions in various quarters and various justifications. We have a moral obligation to protect life on our planet. The aesthetics of life, that's no problem with biodiversity scientists. We love the organisms around us. But this hasn't led to humanity rushing to put large amounts of money to protect life on our planet. There have been calls and links to planetary sustainability. These are the 9 axes from the Stockholm Resilience Center, the 9 parameters that we really should be probing deeply to protect the sustainability of our planet. Biodiversity is one of them, but many of them are not been looked at in any detail whatsoever. Why press hard on biodiversity when it's one of 9 pieces in a puzzle? I think we need to move away from, if we're thinking about this grand project and the grand challenge that, Braulio, you've asked us to consider, we're not going to do it through these very targeted justifications. I think we need to think about the other people that play in big science space. They recognize that we're a curious species. Knowledge is what motivates humanity. We invest billions of dollars as a species every year to understand the world around us. The New Horizons project just flew by Pluto. It cost $700 million to glimpse that planet. I love seeing that image. I think you probably love seeing that image. Stephen Hawking justified it. We explore because we're human and we want to know. Of course we do. It's us. The loss of species is an irrevocable loss of information. Don't let the dodo fool you. Big organisms leave bones, and you can mine those bones, and you can understand that species. But when most of the species on this planet disappear, they leave a smudge of DNA on the forest floor that's gone a day or two later. The loss of information is staggering. The longest book ever written by humanity is Zettelstrom, single volume, 7 million characters. Our genome, the book of humanity, 7 billion characters, 1,000 times longer than the longest book that a human ever wrote. And of course, it's not the biggest genome on the planet. The lungfish, the canopy plant, massive. We love knowledge. We don't burn books. We protect them. The U.S. National Congress Library, $600 million per year to protect information that the citizens of the United States feel is important to humanity. $600 million per year. Right now, all around our planet, this is what's happening. We're burning the books of life without regard, without ever having read them. We need to do something about that. We need to absolutely, as part of a big biodiversity science project, make sure that the books of life are not burned. We need to do this. We need to gather DNA extracts from every species on our planet, perhaps a few representatives, a DNA arc, a library of life, and those extracts are very compact. You can fit 25 million of them in a single shipping container, and we're not going to be able to read those books today. It's too expensive. But we can put those books into archival storage. And some of you may feel this is a bit of a depressing approach. There's life on our planet in a shipping container. But the community we work in, the biodiversity science community, doesn't just grab DNA extracts. We will collect organisms, we will archive those organisms. Each of those DNA extracts will be connected to a specimen whose phenotype we can understand. It's not good enough just to read sequences. We can't reconstruct that organism phenotype. So this is an important part of the work that we can carry out. What are we going to do with all these DNA extracts? I don't like the idea of a shipping container. I like the idea of the model that Norway has created for a very important segment of life on our planet, the global seed vault on Svalbard. We've got agreement to move the most important plants on our planet into storage in this particular facility. Norway's done a good job of it. So I like the idea of them building a second tower on Svalbard, and the global library of life will reside there. And it will reside in other places as well around our planet. But this would be a nice symbolic gesture, so I'm counting on Norway to build that second tower. Now, Braulio has mentioned, and Malcolm mentioned, the fact that we don't understand the number of species on our planet. And how can we fill up that vault on Svalbard if we don't know how many things? The titles of the book, the index, how are we going to do that? And again, as Malcolm mentioned, and you all know, about 1.7 million species on our planet have been named over the last 250 years, and the guess is there may be 8.7 million. So there's a lot of work left to be done. Animal life is thought to be the largest compartment of biological diversity when you're talking at a species level. What would it cost to do the animal kingdom? And we have an estimate of the cost using traditional taxonomic approaches. It's $250 billion to describe 5 million species. And Braulio, you understand big numbers, but I'm pretty sure you also understand that when you talk $250 billion, that just means that science project is canceled. It will never be done, ever. Humanity doesn't spend that much to satisfy its desire to understand. The smart, big science communities understand the top amount of money you can ask for is about $10 billion. Large Hadron, Hubble telescope, that's how much we're invested in them. We need to take that $250 billion and drop it 2 orders of magnitude, $2.5 billion to complete the inventory of life on our planet, and a lot of other things as well. How are we going to get there? George Hale wrote to the Rockefeller Foundation in 1928, just before the Depression set in, and he pointed out these two very important things about scientific progress. The way you move science is with technology and new approaches. And there are times that you can do this, and there are other times that you can't. And in our science, we're perfectly positioned to follow Hale's maxim. We've got new technology. Sequencing technology is moving like a rocket. Sanger, next gen, and next gen, we're going to be able to do absolutely incredible things on this front. And then we have this simple little conceptual advance that says you can tell these objects this we're interested in by reading little bits of DNA. We don't need to read entire genomes. It's too expensive to read the genomes. We need to index life with barcodes across the spectrum of, initially, eukaryotes. This kind of project begins to move biodiversity science into mega science, the billion-dollar-plus projects. There have been 22 of them on our planet, and they all follow a fairly formulaic approach. There's a need for a scientific plan with very crystal clear goals that are big, insightful, and that's really, I think, one of the most important things to capture this money. The other thing is we need a management structure. All projects have a well-defined management structure. They're always international. They are a long duration. People like myself that might be around at the start of them usually, or trying to get them started, usually aren't around to see them actually activated. Hale never saw the telescope on Palomar. He was dead, long gone. So don't worry about a conflict of interest when these old guys like me suggest a project. We're not going to be here to spend the money. You young guys will. Long duration, multi-decadal, and solidarity. And that's something our community finds very hard to do. Get solidly behind an idea and not shoot inwards as opposed to outwards. I don't think we should be shooting outwards either. I just think we should be immensely supportive of our community and the various kinds of science they want to do. But the science that we're doing won't wait. This year, 3 mega science projects were launched. All 3 involve the construction of a large telescope, and they follow this pattern of somewhere between 7 and 10 nations, and the nations rally up and invest somewhere between $100 million and $250 million in these projects. 3 big telescope projects. We haven't been playing in this space. So let's look at one of the big projects and ask what they've done. The Hubble. It's been in operation for 25 years, and this is the publication volume from the Hubble Space Telescope. It rose rather quickly to about 700 publications per year, and it slowly slid up to about 800. That's what happens when you spend $10 billion. This is what happens when you spend much less than $10 billion, and it's barcoders involved. The number of publications has passed the Hubble, and the number of citations actually is about the same as the Hubble papers are acquiring. So we're not completely divorced from, we've not been in mega science, but I would say we're in mesoscience. We're working on a scale that's allowed us to learn some of the ways of collaborating and cooperating that can make it possible to dream big. 2007, we were sitting in this room when we thought about launching the International Barcode of Life project, and we said, hey, in 5 years, if someone would give us $100 million, we will barcode 500,000 species. It took us 3 years to get organized and get the money, enough of the money, to have the courage to say, okay, we're going to go for it, and there's a high risk we may get embarrassed, but we're going to try. And so we launched in late September of 2010, and since then, many of you in this room have contributed to this giant collaborative project. And guess what? These little dots show where barcodes are, and that was 2010. That was 2013. That's 2015. The barcode records have risen, and what about that target of 500,000 species? Guess what? We passed it last week. We're over 500,000. 500,418, I think it was when I looked. And honestly, I didn't quite believe it, so I yelled at Sajeevan and said, don't tell me you're fudging the results on BOLD. He promises me, he didn't. Okay, so we've made that, iBOL, you in this room have made it possible for us to reach this target. But we've only begun. It's 5%, 10% of the species on our planet. What have we learned? We've learned that we can collect organisms. We can collect them in various places around the world. We've found that some of the earlier objections is we're not going to be able to get the specimens to analyze. We satisfied that problem in spades. We could have had tens of millions of specimens if we'd had the resources to run them. We'd never done a big sequencing facility in our community. 5,000, 10,000 specimens per year was sort of thought to be rather good. The little facility that many of you have seen, that small building over there at Bio, ran 1 million specimens through the analytical chain last year. 1 million in a single year. That's been my grand goal for a few years, to see if we could hit 1 million. It required working 6 days a week and some really fantastic people who worked there, but we did it. So we can run millions of specimens. We needed an informatics platform, and that has been assembled with the help and advice of people around the world. So we have somewhere to store the data, we know how to collect the specimens, we know how to sequence them. What we don't know, can we barcode the planet? Can we build this comprehensive reference library, Braulio? And one of the problems is we don't know how big the job is. It's 10 million, it's 20 million. But what we have been doing over the last few years is working in Canada to ask, can we barcode all species in Canada? Now, you've seen Guelph. Canada's a big country. It covers 7% of our planet's land surface. So it allows you to get a little bit of a sense of how difficult it would be to sample in different places. Biodiversity, we're not Brazil. We're 1% of the world, but in many, many studies that have been looked at in different groups of organisms, Canada hosts about 1% of the species on the planet. So if you could do it in Canada and then you can do it 100 times around the world, you've done the world. So, what do we know about Canada? We know that there are about 120,000 species in our nation when the work began, about 10,000 species of plants, 85,000 animals. And these are the groups that we focused on because their taxonomy is best. We wanted to ask, can we finish them? The plants, vascular plants are done. And one of the reasons vascular plants are done is because herbarium sheets were just an excellent source of DNA, and we could rapidly drive through the plants. So I see no problem doing the plants of the world. There's only 400,000 plus species. Non-vasculars we will have done by 2018. So when you walk out into nature in Canada by 2018, plants are going to be under control. Animals, we encountered a few more barriers. One of them is there are many, many undescribed species in our country. Our taxonomic capacity is declining, so we had real problems identifying specimens, despite some help from friends. And the other problem is sequence recovery. The animal specimens, a lot of them were old or they were in formalin. So really we had to think about launching a new collection program. And to solve this thing, we had to figure out how we could process lots of things where taxonomy was not in place. The taxonomic problem was solved in the animal kingdom by moving towards a DNA-based system, looking at sequence clusters. And here you can see some standard sort of barcode sequence clusters, and connecting this with the bin system, where all of those clusters are assigned a persistent identifier, and now bold as each sequence enters bold. It's an animal sequence. If it meets the quality standards, it gets assigned to a bin. And there are, as of later next week, 400, and now it's about 419,000 pages like this that assemble specimen information, distributional information, images of each of these species, the 420,000. And I say species, even though they're sequence clusters, because there's a wonderful mapping of bins onto species. So here's Canada, a bunch of groups. Rosemary, you won't like to see this, or maybe you will like to see the fact that the spiders, those are the spiders that are sitting up there above the line. I don't like them. The others are sitting on the line rather nicely, but we actually think there are quite a few cryptic species of spiders not recognized by current taxonomy. what happens in Canada happens in other places in the world. There's a very tight correspondence between species counts and bin counts. It's the automation of this one really important axis of biodiversity measurement. We can now automate the counting of animal species anywhere on our planet simply by gathering a little CO1 sequence and slamming it into the Russell algorithm that assigns and creates bins. So we now can count, and this has been invaluable for our work in Canada. But the automation is not yet complete. There are other things that we can do, and one of them is we can ask how many species, what fraction of the species are shared between sites. Just press a button, and you can see that 1% of the species are shared between Costa Rica and Ontario. You can see that 18% of the species are shared between Costa Rica and northern Argentina. The automation of biodiversity science, this is where we must head. The other thing is it's not complete yet. There's a lot of phylogenetic signal in the CO1 sequences, and I don't have time to look at this in much detail, but here you can look at two rather closely allied insect families. These are circularized neighbor joining trees. Green are ichneumonids, red are braconids. You get this perfect separation of these two insect families based simply on CO1 sequence information. And that's true for family after family. The taxonomic community has built a wonderful skeleton of biological diversity on our planet, and now we can use that to place unknown taxa. So this is where we're headed. You go out into nature, you collect a specimen, you sequence that one little gene region, and you can place it to a phylum. You can place it to a class. You can place it to an order. You can place it to a family. And in some cases where taxonomy is complete, you'll place it to a species, a Linnaean species, but in lots of others, the terminal point will be a bin. The automation of this critical aspect of biodiversity science made it possible for us to go out and look at biodiversity in Canada in an extensive way. What about recollecting species? I said that was a problem. We have 30,000 species of insects known in Canada, just 30,000. It's thought there may be 55,000 species. So that was our target. How tough is it to go out and recollect 30,000 species of insects? Going to take you forever? We decided it was important to get school kids involved in this, so we sent out a Malaise trap earlier this year to 68 schools across Canada at those sites and said, kids, collect bugs for 3 weeks, send them to us, and we're going to barcode about 15,000 of them for you. And when we did that, we found the school kids had collected nearly 3,000 bins. They collected 10% of the insects in 3 weeks. And guess what? They were grade 5 kids. So I don't believe anywhere on the planet we can't do this kind of sampling and get a good indication of species. And then you don't need to look at a whole bunch of places. You can come to my backyard, as we did, set up a single Malaise trap in my backyard for a couple of years and ran just a fraction of the specimens. And I share my backyard with 4,700 insect species, 16% of our known fauna. And we went to our national parks and we set up a Malaise trap in each of the 45 national parks just for one summer, and then ran about 800,000 specimens, 20,000 per park. And we did that. There's the known species. There's the bin count from this little national park survey. We collected 133% more species than are known from our country. And when you put all this together, the various sampling sites, 29,000 species known, we've now collected nearly twice that number from sites in Canada that we have sampled over basically just a 3-year period. We can rapidly make fresh collections and inventory life on this planet. On animal species, we're up now, not just insects, of course, we've been analyzing, we're up at about 40% more than the number of known species. So I take these results as very good indication that we can actually get a lot done relatively quickly. And Bob, I'm really conscious of the time. Shall I just jump right through these things and not talk about them?
I think the audience would prefer to hear your perspective.
Are you sure? All right, otherwise all right, so I'm going to try and rush through these things. But we're working in Canada and we're trying to find out, and the question you want to ask yourself, of course, is does this help inform something larger? Does it help to tell us about how accurate the counts are of animal species on our planet? the forces limiting diversity. How old are species? There's a nice paper in PNAS just earlier this year by Nigel Stork and colleagues that said, look, if you want to understand animal species on the planet, you need to count beetles, because beetles are almost all animal species, and almost all animal species are insects. So just count beetles. And the estimate they came up with was 1.5 million beetle species, and there are 5.5 million insect species on our planet. This is sort of an important question to answer. How accurate is it? They're pretty sure it's quite accurate. But we've been doing this in Canada, and Canada has 1% of the animal species. Here's the log of the known species, and these are the log of the bins. And you can see that almost, these are 30 insect orders, 28 insect orders. Most of them fall under the line. We only collected at a few places. We did not collect all of the species known from Canada. No surprise. This is a log scale you may note here. Diptera and Hymenoptera fell way above the line of known species in Canada. And in fact, if you do a log normal prediction, 7,000 species of flies known in Canada, we've got 36,000 projected now. A gross, far, far, far more fly species in Canada than beetles, that's for certain. And far, far more than we had anticipated. Those flies, did you find crazy diversity in all fly families? That sort of interests me. Dan hates this kind of science. I just love it, but Dan just hates me for wanting to count species, and I'm always trying to convince him it's worthwhile. I probably will fail again in the morning. There's the regression between number of known species and the number of bins observed. A nice tight correlation with two exceptions. The sesiomyids and sciurids have gone crazy. Isn't it interesting? They've got an unusual breeding system. They're haplodiploid. And in fact, when we sit back and look at sesiomyids, we've already observed 8,000. We've run 90,000 specimens, and the number of bins based on a log norm model is 15,400 at the handful of sites, the 45 or 50 sites that we've sampled. That many species. Based on the insect work that we've done in Canada, we're now estimating there are 95,000 species of insects in Canada. There were 25,000 known, and prior work had estimated there might be 55,000. And so we've just about doubled the known species count of insects in Canada. And if you place this in a global context, back to the old 1% rule, there are 9.4 million species on our planet, not the 5.5 million that Nigel Stork had estimated. Sesiomyids, we're now estimating 20,000 species of sesiomyids. Prior work was a 1,000 species in our nation. And you put that in a global context, 20,000 species in Canada, 2 million species of sesiomyids. What are the implications? Well, the one thing we've got to do is go back and tell Haldane he was wrong. God does not have an inordinate fondness for beetles. He does for stars. They're 10 to the 23rd stars. But beetles, nope. On further thought, stars and midges. And in fact, I'm not prepared to jump quite that far yet because, as Mark Blackster will certainly remind me, we don't have any clue how many species of harpacticoid copepods we have on our planet. We don't have any idea how many nematodes there are. Mites, it may well be that these are the really hyper-diverse groups on our planet. So we can't estimate species. These efforts to estimate species without data, you need to come out and look at these hyper-diverse lineages before you come up with species counts for our planet. What is the implications of this? I mentioned earlier, of course, the people have been down this road before, and one of the people who have been down this road is the astronomy community. Naming conventions. We used to name stars until Mount Palomar opened up the heavens, and what did astronomers do? There are too many stars to name. So they moved away from naming things to moving them into a coordinate system. And I'm certain that as we begin to reveal these immense pools of biological diversity, unnamed biological diversity, we need to think about a new naming model. The fact that we're probably going to be, this is going to be the terminal state. No one's going to pay to name 2 million species of sesiomyids if that number proves accurate. But this is very functional. What limits the number of species? There have been two competing hypotheses. One is competition, and the other is it's damn difficult to speciate, and so you're going to have, there'd be a species under every bush if it was easy. And I'm thinking that when we see these explosions of diversity in haplodiploid organisms, that this is telling us something about the factors that regulate species numbers, animal diversity on our planet. Dan, it is important to do this work. Species ages. We love understanding speciation. The cichlid fishes of Africa, these model systems of very young groups are very attractive as we probe evolutionary patterns. But our science, evolution, is based on case studies. Whoa, what the hell is that? Oh, that's a regression line with an R² of 0. The one, we're now working on continental scales, not with a single group of fish or the single beetle genus. We're working on every genus, every species on the continent, and guess what? There's the mean nearest neighbor distance. There's the number of congeneric species. The cichlid fishes are out here. 0 divergence. And in North American Lepidoptera, 1,100 genera looked at, those are the cichlids. Those are the things with no divergence. The species that we share our planet with are extremely old. 6.27% average nearest neighbor distance, about 3 million years old on average. So, are estimates of global number of species accurate? No. We're overlooking diversity in these groups that no one has studied. Has competition been the primary force in limiting number of animal species? I think we need to give thought to the fact that genetic factors may be playing a really prominent role. Are most animal species old? Yes, they're worth saving. Can we barcode the world? I think so. In Canada, we've done these groups. We've got work left to be done. It took us 1.5 million sequences to do this. I think we can finish Canada for another 1.5 million. If Canada's 1.1% of the world's biodiversity, this is what we need to do. These are the sequence targets. How will we get there? We need to create a massive mega science project, the Planetary Biodiversity Mission, which is going to probe species with DNA barcodes, but that's only the beginning. That helps us get to the inventory. An inventory of species coupled with ecology, evolution, and morphology of these organisms. A library of life up to Svalbard, all of those DNA extracts stored for future genomic analysis. A big effort on advanced biosurveillance that allows us to read biodiversity anywhere on the planet with ease. What do we do now? 2016 to 20, we clarify scientific goals, build support, secure the funding, a 20-year project, and we can complete this. Probably you will have it by 2040 if you get us the $2.5 billion. This room, archival nodes we need to collect around the planet. These are the nations that are represented in this room today. There's no better recruitment center for the collection program that needs to be associated with this project. We need to sequence them. I think we need to think about creating biotrons, big analytical facilities that are all analyzing about 1 million specimens per year. You do this for 20 years and you've analyzed the 300 million specimens. We've got an informatics platform. No problem to expand it. I think this is an important, really important thing for us to think about. The people in this room are taxonomy, and we've tried to spread things out. Past barcode meetings have been pretty much the biodiversity science community, but we felt it's really important to this meeting to bring in the disciplines. If we're going to launch this project, we have to have the smartest thinkers in ecology, evolution, people who understand the technologies, and people understanding genomics. This can't be just a biodiversity science project. It must be very broadly based. We need linkages with the key organizations around our planet that help and focus on biodiversity issues. So, my final slide, you'll be happy to know. We live at a time when life on our planet is threatened. We live at a time when technology is making it possible for us to read life on our planet. We've shown that we can work together as a community. Canada's been a useful pilot project, and this is what we need to do over the next 20 years to get us much closer to protecting the species that we share, are embedded with in this final frontier. The work I've talked about is only due to the fact that there are 243 magnificent people that have worked at BIO. This Wordle, the size, number of years they've worked at BIO, it's way too cold. I don't like it. I mean, these people are very passionate about what they're doing, and so I would like to see some hotter image, but I can't figure a way to present them. I deeply appreciate their commitment to our work. And of course, the work would have been impossible without funders in Canada and colleagues around the world